Good. That's what I wanted to hear. Lovely. So Lila just shared the link for the paper today, which is fantastic. I'm just going to copy that and put it over on YouTube so people there can also follow the discussion. Wow, stop it. Too quick. It's Monday morning. Slow down. As you can see, we're going to have a journal club today on a bioarchive preprint paper that Lilith will present to us. The rule of the game is that anyone is welcome to present a paper that they thought was interesting, and basically just come online and tell us why you thought it was interesting, and then we discuss it to the best of our knowledge. Just some admin before we get started. I know she's keen to get into the paper. We have 4 more Neuroanatomy. This is one of them. And we have 1 more CNS talk this month. And then we're going to take a winter break in December. And we'll be back on the 9th of January, I think, but it'll be in the newsletter. So watch the space. Right, Lilith, without further ado, take it away.
Yeah, hello. As you can see, I'm super excited about this paper. I want to share you this one. Well, this paper is very popular right now, I think, on Twitter. And it was even quoted by someone as the biggest discovery in neuroscience and probably the biggest ever in neuroimaging. So it will be nice, I don't know, to discuss after if, in your opinion, this paper deserves such a title or not. But as for me, this is very exciting and mind-blowing paper because basically we were taught, everyone were taught that we have a homunculus in primary motor cortex, and it is organized in a specific field, in a specific way. And it was the case for around 100 years. And I think all professors, you have to change your slides right now because it feels like that's not the case anymore. And these people challenged that idea that was obvious for most of the people. And this is their new model. As you can see on the left side is the classic Penfield homunculus, and it is organized in a linear way, so from toes to face. On the right, you see the new model that they provided, and there are 2 main differences. So the first one is the organization. They challenged the linear organization, and they said that actually our body in primary motor cortex is represented in concentric functional zones, like functional rings. So what is that? So if we look, for example, at this picture, this is a part of primary motor cortex, and if we look, for example, at this part, we see that there is activation, only 1 peak for toes. But around toes, there is activation of ankle. So what would Penfield say? Penfield would say that we have activation of toe, then we have ankle, knee, glutes, and everything. Which is true, but this is not the entire picture, because we will have the activation of ankle not only below, but also above, but also on the left, but also on the right. So it's like not linear way. We are represented, our body is represented not in a linear way as Penfield told, but in a concentric rings. And throughout whole primary motor cortex, we have big 3 peaks like toes, and then we have those rings around, and then we have the second one is a hand, and we have all these rings, and then the third peak is a tongue, and then we have rings around the tongue. So here we have it: toe, around, hand, and tongue. So this is the first thing that kind of shatters this linear representation of primary motor cortex. And the second thing is these parts, these little dolls, and they call these parts in primary motor cortex as inter-effector. 1 second. It's for the later. Yeah, and this inter-specific inter-effector regions. So first of all, these regions were not included in the Penfold's model. And second, what are these regions? So if we look, for example, at this plot, what they did, they put 6 positions. This is primary motor cortex, precentral gyrus, and they have 6 points. These points represent seeds. So if we put the seed here, we will have like connectivity just in one part of the brain. If we put on the 3, we will have another bulb. And if we put on the 5, we will have another activation there. So this is consistent with Penfold's theory, and these are represent effector-specific regions, so hand, face, and feet. But if we put on 2, 4, and 6 our seeds, we will see that there is a network that activates simultaneously. So let's say if we put the seed on 2, there will be activation not only in that region, but there will be activation also on these other regions that are located between effector-specific regions. And if this is the case, if we put the seeds on each of these locations. So basically, they found a distributed network within primary motor cortex. And this result, they found this not only on their data, but they also validated this. They found the same network on already collected dataset previously, the huge datasets like a Human Connectome project, UK Biobank, and they said that they found this effect both on group level, but also at the individual level. So this is the second main difference from Penfold's model. And what else they found? They found that these inter-effector regions, they have higher connectivity with this system, which is CON. It's a Cingulo-opercular network. And Cingulo-opercular network is critical for execution, action, physiological control, arousal, processing of errors, and pain. So there is a network within primary motor cortex that that has stronger connectivity, functional connectivity with this higher order generic network than effector-specific regions. So this is also crazy, I think. Another thing which is also kind of surprised me is that structurally speaking, we know that motor cortex is I thought it is the most thickest region in our cortex, and it is homogeneous, but it appears so that that's not the case. And those inter-effector inter-effector regions. They are thinner than regions for foot, hand, and mouth, and they are almost as thin as cells inside a prefrontal region. Yeah, so that's also interesting. What else? Another, yeah, and so they asked what is the function of these inter-effector regions, what they serve for. So they did an experiment where the task involved action planning and action execution, and they found that those regions were inter-effector regions. They were active more during planning than execution. So in primary motor cortex, there is a region that is has higher activation during the planning than the execution itself. Yes, and yeah, I mean, this paper is so informative. There are so many different things they found. They also tried to find this network in infants and even neonatal children. Sorry, I don't know how to call them. Yeah, so they didn't find. They found them only in children with 7 months. I don't know if you can see this. No, I don't think so. Yeah, so they found that in 11 months, not 7, I think. Yeah, they found it in 11 months, but not earlier. And then, yeah, so it means that there is an adult-like network already that early in children. And another thing, they tried to compare it with monkey brains. So actually, in non-human primate research, it was already known. Already, they found some evidence that primary motor cortex is not as straightforward as Penfield put it. So they divided primary motor cortex in monkeys in two parts. The anterior part, which is more for gross motor movements and like body movements, while posterior part is more for fine motor movements. So and they found that the anterior motor cortex is more similar to those inter-specific regions in humans. Yes. I think that's all I wanted to say. The only thing that just I wanted maybe to finish this. Well, 2 days ago, we went to a movie festival in Nijmegen, and there were like documentaries about different scientists. And one of them that we watched was on Noam Chomsky. And he said that like cognitive psychology, cognitive science is at the very, very early stage of development. Like we haven't even reached like kind of Newtonian, Galileo level of research. And we're super early, and we need to ask obvious questions that are obvious for us, like why if we throw away the ball, it will fall down, why the steam goes up. And basically, I think these scientists, these researchers, they asked an obvious question that was super obvious for everyone else for 100 years, more or less. I mean, in monkey brain, they did some new research that was giving hints that that's not true. Yeah, but that's, I don't know. I really like this paper. I think it's breakthrough, and it will open new research that is connected with somehow with motor functioning and motor cortex. Yes, what do you think?
Thank you, Lilith. This is indeed an exciting paper that caused a lot of buzz on Twitter before everyone left Twitter. Oh, hands are going up. Here we go. Victor, you're first.
Nice. Thank you, Lilith, for the presentation. Just a quick question. Can you go back to the figure with the activation of like different parts of the part, no, not this one, like the true activation of the connectivity here of the motor cortex, where you can see like there is like concentric activation.
Okay. Yes.
Perfect. I was wondering on the figure A on the top left, you were saying that they were concentric, so it was only the toes in the middle, then around the ankle, for example, then around the knee. But I don't exactly know how they made this map, and I'm wondering if actually the ankle also encompass the toe activation and the knee encompass the whole, and actually it's like more of a kind of hierarchy and than a concentric activation. Do you have more information about that?
You mean like if we need to, let's say, to move ankle, we will move toes and ankle? You mean that?
Something like that, that actually the activation encompass the toes, the ankle activation encompass the toe, and it's maybe a bit more complicated than just
Yep. I didn't go so much in details how exactly they've mapped it, but yeah, there are some details about that they use Gaussian something. I don't know. I feel like it's better for you. I mean, if you want to know.
But actually, I think figure B is somewhat answering my question, because you can see the profile of activation with that score. And in light blue, you can see if it's the same color code.
Yeah. So this one is hand, and around should be wrist. But to be honest, it's not so much around it. Like
but you can still see that there is a deep.
There are two peaks. So that's what.
So it's apparent that it's quite more complicated than what we may have thought, and that they really are showing something interesting here.
Yeah, because here, for example, I don't know if you see the mouse here, there is activation as well, but not so
strong,
high as around. So maybe when we move the toe, we have also a bit of activation of. Oh no, if we move the hand, we have a bit of activation of also shoulder, which kind of makes sense maybe.
Yeah.
We are not so isolated robots. Yeah.
All right. Thank you.
There was a question in the chat. Let me read it out to you. This brings up the question of the relationship between activation and observed behavior. Is this new motor homunculus really motor or representational?
But why motor? But what's the difference exactly between motor and representational? I mean, the Penfield, he showed the representation of motor homunculus, right? So I think they are showing as well a representation of motor homunculus.
I'm back. I'm sorry, I couldn't ask it directly because I had to finish something, but that's fine. So Penfield used to put an electrode and see directly a movement of the arm. Here it's different. You ask participant to move that finger or tongue, and And we know we can activate the motor cortex just by imagining that you move your toe, your arm, or your tongue. So the difference is, it's contrasting the two is interesting, but there is something more that you're measuring when you ask people to move rather than using an electrode and making them move independently of any other processes that is in the brain. And so that's why I asked the question, or maybe that's why there is a difference here.
Yeah, then there should be additional research with but I don't know if that's possible, but maybe with open surgeries when it is allowed to do that. They can, test maybe not the whole primary motor cortex, but some part, and see if that's the case. And it will give like additional evidence that probably this also at the same level of evidence as Penfield's homunculus. But the thing is also Penfield wasn't saying that this picture is a perfect representation. I think they cited him. They said something like, they said that they cited an ace to memory cartoon of representation in which scientists' accuracy is impossible. So he also know that this is kind of messy. So, and it was just a simplified version to allow us to remember more or less what's located. But for sure, the thing that he didn't find is this inter-effector network. Yeah. But yeah, that's a cool point that we cannot really compare these two models for now.
Michelle has another question. Just go ahead.
So sorry about that. As I'm getting older, I'm becoming a pain in the ass. So sorry about that. So cortical thickness was assessed derived from T1 imaging, isn't it?
Cortical.
T1, T2.
Probably, but I don't exactly say that. It's very compressed, and in supplementary materials there are just videos.
Probably no imaging. So I learned that the hard way and got shut out by a world expert on cortical thickness. So the cortical thickness of the motor cortex is not well estimated from T1 due to the high myelination of the first layers. So it is actually supposed to be 5 millimeters to 6 millimeters, but when you do it with neuroimaging, you only have 3 millimeters because you just pick up the top layers of the cortex and not the lower layer of the cortex. And so I think if that person, which I named in the chat, was a review of this paper, I'd be really upset to see that.
Okay. Yeah, but
that difference only applies to the motor cortex or other areas as well?
So any areas that are high myelination there is actually a correlation between myelination and the evaluation of cortical thickness in the cortex derived from neuroimaging. So any areas that have high myelination tend to reduce cortical thickness. If you change your myelination, you tend to reduce or increase virtually your cortical thickness. And the myelination is more or less different according to the different part of the brain. If you were to draw a correlation between postmortem measurement of cortical thickness and in vivo measurement of cortical thickness with T1, I don't think it would be significant. I'll let you draw your conclusions from that.
What a cliffhanger. I actually, gosh, I'm going to be that annoying person, but I have a comment, maybe a question. So listening to you describe the paper, I actually was reminded of Marco Catani's paper in 2017 in Brain, where he remapped the homunculus and he actually offered a new map of what the original homunculus should have looked like, given that it was a schematic representation and not scientifically the most accurate, as you just have shown us. And he also tried to remap the original stimulation points from Penfield, and actually showed that there is quite a large overlap between the individual areas, and it's not as clear cut as the classical drawing of the homunculus on the motor cortex would make you think it was. So I just wanted to throw that in, that actually there is different versions of the homunculus out there that we can compare it to. And I'm just going to do a very quick search, so I'm not making this up, but yeah, no, it's not actually coming up. I'm not going to put that on record, but I remember there was a discussion about the female homunculus and if that would actually look different compared to the homunculus that we know. But yeah, so that paper was 2017 in Brain, so it might be interesting to compare the different homunculi that we have.
Yeah.
Sorry, no question, but
yeah. The only thing I wanted to say, the last thing maybe, or I don't know if there are questions, like they are calling these regions mind-body interface. I don't know, it's kind of reminds me Descartes thing, but I'm not sure if that's the correct way, if it is too exaggerated way to name this region. But yeah, this is the only thing that kind of made me to be suspicious about. But yeah, other than that, it's very cool paper, I think. Yeah.
So as you mentioned, it was super hyped on Twitter, and you introduced it as potentially the most important paper for neurosciences or neuroimaging. What are the things that will change now that we have new maps?
Sorry, my connection was bad. Could you repeat the question?
Sure. So you introduced the paper as one of the most important in neurosciences or neuroimaging.
Mm-hmm.
My question to you is, what's going to change now that we have the new maps?
Yeah, well, I think it's we can think about like people, I think, will start to try to understand exactly what are those regions are actually doing, and if there is probably some influence of this network in recovery of patients, or if this region is connected somehow with some neurodegenerative diseases like Parkinson, or some diseases where action planning is compromised. So maybe this network plays an important role there, but I don't know. I feel like people will not ignore anymore that and start to ask questions if this network is doing something very important in motor movements. Yeah, so yes, I don't know, but I think they will consider this in future research, all the researchers that are connected with motor research on motor cortex and motor functioning. Yeah. And also the other thing, it's 10, but okay. Yeah, the other thing is that, I mean, a visual cortex also kind of represented in a concentric form. Like, let's say if we take an eye, right? I mean, it's not cortex, but like eye also, we have the fovea, and then we have eccentricity that goes like further. So, and it's nice to see that the primary regions, like the early level of processings in our brain, is represented in concentric forms, also in a motor cortex. I don't know, this was also a nice idea.
Final question from Anna.
Not a question, just a follow-up on your question. What is exciting about it? I would also say, or at least suggest to study inter-individual differences in a very much more detailed way. For example, looking back at the, I'm hearing myself double. Looking back, for example, at what.
We have an interference. I can't hear you anymore.
Yeah, but we don't hear you well.
Victor, you're in the same room. Can you mute your mic?
Oh, but am I not muted? Oh, damn. Oh, sorry.
No, you aren't.
I thought I muted myself afterwards. I forgot. Sorry.
Here we go. Try again.
Oh, good. No, what I was about to say was, to study inter-individual differences in a very much more detail. So the graph that we looked at while Victor was asking the question about with the activation and the Z scores, unfortunately, you're not sharing a screen anymore, Lilith, but there you could see, like, I found it quite interesting that you could see, for example, for I think elbow activation, it was different between the two participants. And I think that allows, for much more detailed insights into very much more detailed movements in different people. So comparing, for example, a healthy population with a clinical population, and then seeing what exactly could maybe drive it. So or which brain parts in detail are responsible for certain differences in the activation maps. So that could be also cool for the future.
Yeah, that's great. Yeah, I agree. It will be really interesting, I think. Thank you.
Lovely. That's all we have time for this morning. Thanks for joining us for the discussion of this fantastic paper. Have an exciting week in science, and I see you next Monday morning. And I think Teresa is presenting next week, right? Yes. Fantastic. See you then. Bye.
Bye.